Okay, I'll go quickly through the seminar list here. So the ESPM seminar on Thursday, 3:30 to 4:30 is going to be presented by Sherry DeVore from Arizona State University. The title is Restorative and Mechanistic Insights on Community to Microscale Biogeochemical Investigations. The IB seminar on tomorrow at 12:30 to 1:30 is going to be presented by Joel Slade from Fresno State University. And the title is Analyzing the Role of MHC Diversity in Immunity Against Malaria and West Nile Virus in Songbirds. Interesting title. Essay Brunch on Friday from 10 to 11 will be presented by Dr. Menglin Wang, a postdoc in the Essig Museum. And the title is Worldwide Historical Biogeography of Termites, Blatodia isoptera. And then Botany Lunch, also on Friday, is being presented by Saskia Rather, if I pronounce that correctly, curatorial associate for algae in the University of Jefferson Herbaria. And the title is Macrophyte Community Structure in an Ocean-Dominated Estuary and Its Implications for Management Responses to Sea Level Rise. We have Herp Group on Monday that's going to be presented by Isaac Crone. And it's going to be a travelogue on our last Sulawesi expedition to 2 mountains and 2 islands. It should be very exciting. Isaac was telling me, how many photos, Isaac? 1,020, something like that?
Far fewer, but I'll try and keep 1,000 of them out of the.
I heard it was a big number. Okay, and then the MPC lunch seminar next week will be presented by Jack Sang, who's a professor here in IB, and his title is Jaws of a Kind, the Biomechanical Consequences of Being Mammoth. Okay, so those are the seminars. Any other seminars that should be mentioned?
I just want to check the Zoom.
Make sure that Zoom is happening.
Okay, I think you're good. I just lost the green window.
So I can hit the forward button here. Okay, so I'm going to provide Ben's introduction, of course. I mean, this is a finishing talk. Obviously, he's already Dr. Ben Karin, so he's already, the main event has already transpired, and this is really the celebration, the finishing talk. And so we get to hear him summarize the result of all of his efforts. And so I don't really have a very congealed intro here, but I just wanted to show you some pictures of Ben and tell you a little bit about his history. So he is a skink man extraordinaire, but sometimes we catch him with other things like a hetula. So I first met Ben when he was an undergraduate here. And so he took IB 104, and so I met him at that point, but we didn't really interact that much. Maybe you were on Friday field trips. I can't recall. But in the end, he ended up working with Rory Bowie on a bird project, and so yeah. And so really, the thing that happened that was really instrumental was Alexander Stubbs, who was already another undergraduate member in my lab, who was coming on this expedition in 2011, said, I've got, Ben, right? He took IB 104. He's really great. He'd like to come on the trip. You should let him come on the trip. And I was like, well, Alexander's full of good ideas. And so, sure, Ben can come on the trip. And which was a great moment for herpetology and a bad moment for ornithology. So these are a couple of pictures of Ben on that trip. You can see how young he looks in this picture. He doesn't look that much younger in this one. But do you remember this? Probably not because you were sleeping. That photo is actually from, we were on Sumbawa Island, and we had to drive back from Sumbawa to Bali to get our visas. And we decided, after prepping specimens all day at 11:30 p.m. when we finished, let's just get in the car now and make the 20-hour drive back to Bali. And so this was in the middle of that horrible journey across Sumbawa. This trip had a few kind of memorable moments in it involving Ben in various ways. So, Alexander, I said he had lots of good ideas, but the worst idea he ever had was buying a motorcycle that we were going to drive across the island so that we could access difficult to reach places. This is what happened to Alexander when he tried to ride it. And so we had to put a more competent person on the bike. So I actually love this picture of Ben because he rode the monster across Sumbawa. And you can see we were already starting to tape it together. And by the time we got to East Sumbawa, which included this 8-hour, horrible washboard dirt road across the island, the motorcycle literally had fallen to pieces, and we just had to leave it in a motorcycle shop. But Ben did great work riding that bike. At the end of that expedition, Ben and Alexander made this kind of incredible trip that I still kind of blows my mind. It was in Eastern Indonesia. So here's a map of Indonesia. It was in this part of Indonesia. And, they don't even show any of the islands here because they're tiny little things. But if you blow that up, you can see that there's an archipelago of islands that runs from K up to Seram. And you might think, well, what's the scale of this? It's 275 kilometers of open ocean. And so they decided they were going to take a boat and they were going to survey all those islands along the way. But they were going to go to the furthest most island first and then work their way back. And by the time they got there, their boat died, and then somebody took off with it, and they were kind of left stranded without a boat. But they survived the trip, and in the end, it was convincing enough for Ben to decide that I guess he wanted to do herpetology after all. It was a pretty epic adventure to do that trip. So after Ben graduated, he went to Villanova University for his master's, and most of you probably don't know this, but Villanova is a really famous program for herpetology masters. It's a terminal master's program, and they've just churned out a large number of really high quality graduate students. And so when Ben finished with his work there, working on skinks on Borneo, he applied to come back to Berkeley again, where he was one of the top applicants to the department, received a Berkeley fellowship. And so I was really stoked that Ben decided to come back as a herpetologist and not an ornithologist. So then Ben got involved in our Sulawesi expedition. So he wasn't able to go on the first two because he joined the lab in 2016. But he was able to come in 2017 on our Katapasa expedition. You can see everybody's very excited about being in Indonesia at the beginning of that trip. Here's Ben, like at the beginning of the trip, where he's getting ready to climb up the mountain, and while we were doing some field work along a river, everything's looking good. But this trip is also famous for having kind of a sad outcome for poor Ben, which is when he fell off of a waterfall and dislocated his shoulder and ended up having to leave the trip a little bit early. And it actually cost him as well. He's still smiling. This is like right afterwards, right after he'd managed to climb back up the waterfall and the other waterfalls, basically, to get up to the trail again. He still had a smile on his face. It was pretty amazing that he made it. But it cost the Torempupu trip as well, right? So, I mean, he had a setback. But he was hooked on working on Sulawesi skinks by that point. And so he was back with us on Gunung Dako, which was really the most successful expedition, especially for Ben. Here's Ben with his Lazarus machine, his Jesus fridge. He's going to tell you about the research that he was conducting using that machine. We called it that. I don't know if you're going to tell them. Are you going to tell them? Were you going to mention it? Yeah, it kind of like blew up. It was like boom, and the smoke came out of it, and we're like, okay, I guess the ecophysiology experiment's out, but it just kept working, and so it became the Jesus fridge, and it worked great for the experiments. Another notable experience while we were doing field work, he was able to go. So we did the Dako trip. The next trip was to Ilamata. That trip kind of had a little snafu as well, where we weren't able to go very high on the mountain because of a landslide that cut us off from our expected destination. But one of the cool things that happened, allows me to show you some fun pictures of Ben, was the discovery of this lizard, which is the first Hemiphyllodactylus ever found on the island of Sulawesi. And it was found in Rory's mist net. And it had fallen out of this giant tree. So this giant strangler fig. And so Ben had the great idea that he had climbing gear with him. It's like, let's go up this tree and set sticky traps and see if we can catch more of this Hemiphyllodactylus. And so here he is preparing. A couple of videos of him like showing his technique. Up he goes. And in this one, he's already up there. Gigantic tree, right? Like really amazing. And so he went up there and he set sticky traps. And in the end, he actually caught a lizard. This lizard, which we thought was a new species of Lepinia. It may still be a new species of Lepinia, actually. It definitely looks different than any other Lepinia infralinealata I've ever seen on Sulawesi. But that was really cool. And he also helped me climb up that tree, which was a fantastic experience. But here's Ben doing his thing. Yeah. So I don't really have anything to say about this picture, except that anybody who argues that our field camps are probably like too intense, clearly not. I mean, you look great in pink. Yeah. So I want to just take this moment to say a few words about Ben as a grad student, and even as an undergraduate. I mean, in the whole time that I've known him, I don't think I've ever seen him upset. At least he's never yelled at me. I mean, he's always upbeat. He's super patient, always positive. a really fabulous person to have in a lab, right? Ian, you should be stoked. And he also was, the most productive student I've had in my lab. I mean, it's kind of incredible. I don't want to embarrass any grad students here with this description of his record, but he came to the lab in 2016. He had his first publication on the basis of that trip that he took with Alexander, which was on salinity tolerance in Limnonectes frogs. It was published in 2013. But by the time he arrived here, he had some papers in the mix already from his master's work. So he started basically in earnest publishing papers in 2016. And right now he has 36 publications with a 37th that's in review. He's published 25 papers since 2019. It's kind of incredible, the amount of productivity that's there. And that doesn't even include really the PhD dissertation, right? Which he's going to be telling you about today. So, I mean, super productive, really fantastic grad student and a wonderful person to have in the lab. So with that, I will give you the floor and we get to hear about your fantastic research.
All right, well, thank you so much, Jim, for that introduction. It's great to see all those photos from the field. We've had some amazing times out there. Yeah, I'm really excited to be here today and share all this work I've been doing for several years now, and I love these skinks, and I'm excited to show you all why they're so cool. So, without further ado, let's jump in. So, really, this question, what generates species, is the central theme of my research. And scientists have been studying this for quite some time. We basically know the answer, that species form if lineages spend enough time apart. This could be they're isolated on different regions, or they have adaptive processes that kind of isolate them even in the same environment. But generally, they just need time. And we know that most of biodiversity is generally concentrated in tropical areas around the world, and especially in those areas, it's even more concentrated in mountains and island archipelagos, where species diversification seems to accelerate even more dramatically. And this is because they foster isolation or foster new environments where things can diverge. So my talk today is going to be focusing on these processes and kind of the relative importance of them in generating species diversity. So island archipelagos are natural evolutionary experiments that have fascinated biologists for a long time. It goes back to Darwin's finches on the Galapagos Islands that have evolved these incredible different bill morphologies for eating different food sources, knoll lizards in the Caribbean that have adapted to different parts of a single tree, and tetragnathed spiders on Hawaii. Or these are all great examples of adaptive radiation, where adaptations are really causing are the reason for their diversification. And island archipelagos are really useful systems for studying this because they're discrete units that with separated populations on them. Mountains are also known to drive speciation. So I love this example from the Andes of these Lialemid lizards, Lialemus lizards that have diversified explosively across South America and are especially concentrated in the Andes Mountains. And you can see that their speciation rate in the Andes, specifically here in red, is way far outpacing the rates in other outlying areas. So the mountains are really driving speciation because of all the unique environments found within them. So let's jump into Southeast Asia and Sulawesi. So Jim kind of gave a nice little primer to this part of the world, which I've fallen in love with. This amazing chain of islands between Southeast Asia and Australia. So Sulawesi here is in the center of the map, and it's in the biogeographic region of Wallacea. This region is unique in the world because it's this set of many islands that are between these two big continental shelves, these big source populations. So the Southeast Asian Sunda Shelf is this projection off Southeast Asia that's periodically exposed when sea levels drop, which has happened many times over the last many millions of years. And the fauna of Borneo, for example, is really similar to the rest of Southeast Asia. Australia and New Guinea share a similar fauna for similar reasons. But this area in between, Wallacea, is this kind of transition zone between these two areas. And every island in Wallacea is what we call a deep water island. Anything that got there, any terrestrial organism that got there had to do it by overwater dispersal. So basically everything in there kind of rafted or floated on some vegetation mat to get to these islands. So it's developed this really unique set of organisms there that are found, that are kind of this filter and transition between these two source populations. So we're going to look at Sulawesi today, which you can see is this huge island in the center of Wallacea. And this is about the relative size of Sulawesi compared to California, which I thought was very interesting. It's a big island. It's I think the 11th biggest island in the world, and it's if you count for all its spindly islands, it's about 2/3 the size of California, which is pretty incredible. And you can imagine there's a lot of opportunity for diversification, especially it's right on the equator. The equator goes right through it. It's kind of perfectly set for diversification of organisms. On top of all that, Sulawesi is this incredible, unique paleo archipelago. So it started out many millions of years ago as two sets of these kind of small proto islands, and it went through these series of phases of development. So first, islands would form off the coast and created this archipelago where everything was isolated. Then there is this period where mountains started to form, and around 3 to 4 million years ago, mountains started to form and developed a lot of topography on Sulawesi. And then the final stage, these proto or paleo archipelago started to come together and form the shape of Sulawesi we see today. And it wasn't until the last million years or so, and even less, where it finally formed its present configuration. So this gives really discrete opportunities to study these different diversification processes through time. So we know from other studies that Sulawesi's paleo islands drive speciation. Generally in lowlands, this has been studied really well in lowland species that have diversified across those paleo islands and then remain separated on the different peninsulas of Sulawesi, which we now know from these new geological models are representative of these paleo islands. So the macaques, Sulawesi macaques, and the toads are some classic examples that kind of started to show these regions on Sulawesi. And then Jim's work on Draco flying lizards has really clearly showed that every single species of Draco on Sulawesi are constrained to these paleo islands, and they were really fundamental in creating the diversification there. There's been other work on younger radiations like the tarsiers and these Bonomi's rats that have developed into their own little radiations, but they're really young, 2.5 million years. So they've only really seen kind of the last period of Sulawesi's development. The older radiation, like the Draco and the macaques that I just mentioned, they're older, around 6 million years, but still, like, they didn't arrive and start to radiate until after a lot of the archipelago had formed. And these shrews have also explosively radiated into the mountains, but again, they're younger, relatively young radiation. So today I'm going to be telling you about a radiation that arrived much earlier in the history of Sulawesi. And it gives an opportunity to understand more about how Sulawesi formed and the processes going on there. So as Jim was alluding to, we've done these mountain biodiversity surveys for many years now. I participated in these in Daco, Katapasa, and Ilamata here. But yeah, the team has been doing these surveys for many years now. We've now collectively surveyed all of Sulawesi's paleo islands and up to high elevations on each of these. So this gives like a really powerful data set to really start to look at Sulawesi here. So this is a huge collaborative project with tons of Indonesian scientists and foreign scientists working together on these mountains, sometimes up to 30 plus scientists at a time in these field camps that we set up on these mountains. We're camped out there for weeks at a time. It's pretty incredible what we're able to achieve with all this infrastructure. We have camps in the lowlands and up higher on the mountains, and you can see here this is like a mossy forest up higher on a mountain. And because of all this, we have generators. We can work at night with lighting. We can like charge our equipment. And I was able to run these thermal physiology experiments, which I'll talk about later. These sites are hard to get into sometimes. They're logistically challenging. Here we are driving across all packed up in the back of a truck, driving across a river into one of the sites. So it's a river about 2 feet deep that this truck had to go across because it couldn't go across the little bridge there. So these are logistically challenging. We have to like sometimes cross fast moving streams to get in with all our gear, and then we generally hire 50 plus porters to haul tons of field equipment up these mountains, and it would not be possible without the help of local people to do these projects. So just to kind of give you a little preview of what some of the diversity we find here, just a selection of herps. Here's a Orifrani with egg guarding here, Brachyphorus gliding frogs, Pterodactylus geckos, that Hemiphilodactylus that Jim mentioned, amazing snakes, and these are some of the skinks that I'm going to be telling you about, Sphenomorphis and Skinks. So before we jump into that, I just want to show you what one of these mountains looks like. So let's take a trip to Gunung Dako together. This is up on the northern arm of Sulawesi here, and together with the whole lab, we've been doing these kind of summarizing all the data we've collected from this mountain, and we're gonna be writing papers on all of the mountains across Sulawesi. And you can see here, if you split up we have the frogs on top, lizards in the middle, snakes in the bottom. You can see most of the diversity is concentrated in low to mid elevations. And elevational range is on the x-axis here. So most of the diversity is concentrated in low and mid elevations, and then there's a turnover at these middle elevations around 1,000 plus meters. We start getting these high elevation species, and this is different on every mountain depending on local conditions and what the habitat type is. But generally this pattern holds where we have like lowland species and mid elevation, then high elevation species. So in the frogs, we see several species have made it to high elevation, and the lizards as well have colonized these high elevations. So this gets up to 2,400 meters, these upper frogs. So not Andes, not quite as tall as the Andes, but given the amount of time they've had to get into these habitats, just a few million years since these mountains formed, they've gotten quite high up there. So if we look at the lizards, if we look at the skinks here, the Sphenomorphis and Skinks skinks, so the Sphenomorphine skinks from my title, you can see they really are the groups that make it into the high elevations here. They are among the lizards. There's a few other species that can get into high elevations that are kind of able to basically exist everywhere, but among lizards, these are the ones that do it. So that's why, as we've gone on these surveys, I really wanted to focus my work on this because I wanted to understand the elevational diversification. And you can see on this mountain, there's 5 species of Sphenomorphis and 4 species of Tithyscincus that are elevationally stratified to some extent across this mountain. So it's a really cool study system that we wanted to dive into further. So here's the skinks, the two genera that I'm going to be studying. Sphenomorphis is these medium-sized leaf litter skinks. Sometimes they're up on the base of trees and trying to find little pockets of sun that pop through during the day. There's only 6 species described from Sulawesi. So you can already tell that there's going to be a lot of undescribed diversity here. Tithyscincus are these tiny little skinks. If you know Batrachosteps, the slender salamanders here in California, they're really similar. They're really similar in size and shape and habit to those. So I like to think of them as the Batrachosteps of Sulawesi. But they're tiny. They're deep in the leaf litter. They're hard to find. We generally only find them in pitfall traps when we're doing these surveys. Collectively, we had over 1,000 samples spanning all of Sulawesi's Paleo Islands from these two groups. So a huge data set to start working through. So the main questions I'm going to focus on are: first, did Sulawesi's Paleo Islands drive the diversification of Sphenomorphine skinks? Second, what are the relative roles of overwater dispersal and mountain formation in generating species? And then third, how have species adapted to high elevation environments? So kind of the rest of the talk, I'll be focusing on kind of what I did to answer these questions. So I needed to do a lot of sequencing first. So I'll start out with the mitochondrial screening to kind of figure out the major lineages present. Then the phylogenomics to get a well-resolved species tree of this group. And then finally, the comparative analyses to look at the biogeography and trait evolution in these groups. So let's start with mitochondrial screening. So normally, researchers screen samples by sequencing a single mitochondrial gene. So pretty short little fragment of the mitochondria. But it's 2023 and we have a new technology. And I was like sitting here as a grad student. I just finished my master's, which had a lot of Sanger sequencing. And it's like, could I get the whole mitochondrial genome for the same cost that I would have got done to get all these Sanger genes? And I looked through the literature. There were some methods I could possibly use with long read sequencing, and I was thinking maybe I could do this. I decided to give it a shot. There's a lot of benefits to getting the whole mitochondrial genome, and developed this method to sequence, basically amplify the mitochondrial genome, which is this circular plasmid inside the mitochondria. It's about 17 kb in length, and I PCR'd it in 2 overlapping fragments you can see here in green, and then sequenced, basically amplified them, performed with a adapter tail, and performed an indexing reaction to do this very cost effectively, essentially. And then I tested this on both PacBio and Nanopore technology. I have a lot of collaborators for this project. A lot of them in the field genomics course. We worked on doing this for Nanopore. These 2 undergrads, Celine and Laura, were really critical in running a lot of these PCRs. They ran hundreds of PCRs together. Even when I was away in the field, they were in the lab running PCRs. So I really want to thank them for all their work. The results are I was really happy with. PacBio data, we can see you got great coverage, 1400x coverage. so I think I may have over sequenced a little bit, but yeah, basically you can scale this up dramatically and get a ton more. If you have more samples, you can scale this up, or use just a part of a lane if you wanted. Oxford Nanopore worked as well with around 65x coverage. So both were effective. 96% of the samples covered at least one fragment, so was really able to kind of place each sample in the preliminary tree, and 84. But there was some dropout with like which fragment they recovered. So that's something that could be kind of tweaked going forward with this protocol. And the cost ended up being around $6.73 each for 677 samples. So compare this to Sanger. If you send it off to Barker, you'd pay about $760 after everything. So for one gene, yeah. So we got a lot more data for a little less. And Nanopore, this is the cost for PacBio. So Nanopore was even cheaper. So I was really happy with that. And getting my first look at the tree, I was really excited to see that Sphenomorphis and Tithyscinctus were each diverse radiations on Sulawesi that originated from single dispersal events. from the Sunda Shelf. So there are these monophyletic radiations on Sulawesi. This is the kind of the first look at the data, but I won't go. I'll save most of the story for later. I took about 5 to 10 of each clade for the phylogenomic analyses. So let's move on to the phylogenomics. So well-resolved phylogenetic trees are critical to studying evolution. We call this the species tree, a species level biology that depicts, the kind of the splitting of the lineages through time, or not necessarily through time, but the splitting of the lineages. And this is essential for taxonomy, biogeography, understanding kind of the movement of lineages across changing landscapes, and also studying trait evolution. So you need a really good phylogeny to be able to really study any of this stuff very well in biology. But gene tree discordance can make it difficult to estimate the species tree. Not every gene tree, not every fragment of the genome is going to give the same phylogeny and not match the species tree. So you can see the species tree here has B and C sister, whereas the gene tree has C and D sister. So they can show different aspects of the phylogeny, or different trees. This can be due to incomplete lineage sorting and hybridization, which are real biological processes that people are very interested in studying, and how they apply here. They could be due to gene duplication or loss, systematic error, which is a complicated thing that I'm not going to get into here. And then finally, gene tree estimation error. So if there's not enough phylogenetic signal in each of the genes you're studying, you might get the wrong gene tree just because you can't estimate it very well. So this is most likely with short loci, or those that have slow evolutionary rates. So I set out to try to get at this last problem here. And with my master's advisor, Todd Jackman, and Tony Gamble at Marquette, we developed this data set called RELIC, Rapidly Evolving Long Exon Capture. So the goal of this being to maximize per locus phylogenetic information content while maintaining presence and orthology across evolutionary scales. So basically to give as much phylogenetic information as possible, but to avoid other problems with phylogenetic markers. So criteria being that they should be single exons present in all amnio groups, so they work across mammals and reptiles. They're also present. Many of them are present in amphibians, but some of them start to drop out. And then they're at least 1,500 bases in length. They're more rapidly evolving than RAG1, which is a common phylogenetic marker that's been used for many years, and already provides fairly good gene trees. And finally, they're single copies, so we don't have to worry about gene duplications. And in total, there's 285 long exons in our latest version of RELIC, and targets around 900,000 bases of DNA. You can see, compared to other phylogenetic markers, RELIC, and here in blue, has more informative sites, and you can see here the length distribution of these markers. They get up to from 1,500 all the way up to over 12,000 base pairs in length. So they're extremely long exons, some of these. And if you compare the phylogenetic informativeness, so the ability of these markers to resolve nodes in the phylogeny at different points in time, you can see that RELIC outperformed these other types of data sets dramatically. So they really should be powerful at resolving nodes in the phylogeny. And this is really due to their length. So you can see a strong correlation between alignment length and the maximum phylogenetic informativeness here. So I built a hybrid capture probe set to target these genes. I also targeted a lot of other genes that I won't be talking about today. I'm just going to focus on the RELIC set today. And this big probe set was a work that Simon and Ammon and I all worked together on to kind of pull all these loci from across the genome that could be interesting. So we have this really cool, I'll be kind of working through this down the road, but today I'll just focus on the relic loci. So to kind of summarize the phylogenomic data set I sequenced from 709 mitochondrial samples of Sphenomorphis, I took 282 for hybrid capture, and for Skinks, from 232 samples down to 125. So huge data sets here. And you can see both of these groups are distributed all over Sulawesi. Except for Skinks, we think potentially might not even occur here on the Southwest Peninsula, or we haven't sampled it yet. So I got my first look at the phylogeny after analyzing these data. So I was really excited to see that all but a few nodes received less than 100% support. The phylogenetic tree seems to be very robust with these relic loci. I'm definitely going to be doing further testing just to confirm this down the road, but I was really happy with these results. You can see the Sphenomorphis is split into these 6 major clades, and Skinks into these 5 major clades here. And based on my preliminary results, it looks like there are up to 33 candidate species level lineages in Sphenomorphis and 26 in Skinks. So these are diverse radiations, and I think this is a conservative estimate. I think it's actually probably higher than this, and we probably also haven't sampled every species on the island. So I think as I go forward with formal species delimitation analyses, I'll get a better estimate of the true diversity here. But pretty incredible radiations here. I then had to convert this phylogeny into a time-calibrated species tree. So I used this program called BPP, which is a Bayesian multi-species coalescent analysis. I used a fixed topology and estimated the timing of divergences with an established molecular rate. And I got a times calibrated species tree here, which I'll be running these comparative analyses on. And you can see here there's a point in this tree where there's a lot of nodes kind of stacked up on each other. I've done some analyses since this that have started to tease that apart better. It just seems to be, I seem to have not put enough. I only took 2 samples per lineage in my first pass at this, and I think I just need to run the whole data set. So, and that cleaned things up a lot. So, but it doesn't change the story at all. So I'm going to be presenting today because I have all the figures for this first analysis. So let's move into the comparative analyses. So biogeographic modeling, we can use a Bayesian approach to essentially look at where species occurred through time. And we can do this by splitting up and we can use the information from the geology and the geographic literature to split up the past into these time bins with different land masses availability and different dispersal probabilities for each landmass, so for each area. So I went through these different maps essentially and noted which islands were available, which areas were available, and then essentially gave a different multiplier if it was connected by land or an overwater dispersal, and various other details there. And this modeling approach also allows us to look at different types of speciation events. So I'm splitting these up into within Paleo Island and between Paleo Island speciation. And you can see here that essentially if a species that was originally on an area, let's call it the green central core area, split into 2 lineages that were also on the green central core area, this would be I would call this sympatry range copying speciation, but really it's not sympatric speciation, it just means it's in the area, the sympatric area. So I would denote this as speciation within a peak Paleo Island. Versus between Paleo Islands, there are 2 different options here. Like there is if like say a species was distributed on green and yellow and then divided into a species on yellow, one on green, that would be vicariance. And finally, a founder event would be kind of green going to these offshore islands. Blue here would be dispersal immediately followed by speciation. So these are kind of, I'm not going to go into too much detail here, but those are the kind of the general models that I'm testing. So let's get into the good stuff. This is the biogeographic model across the sphenomorphous phylogeny. And you'll see here, so we first have pies on each of the nodes. I know they're a little small, and I'll just go through it in more detail in a second, but pies on each of the nodes, this corresponding to which paleo island area they're on at different points in time, followed by these little dots are it would be a founder, a sympatry or vicariance event, and then to the right of the phylogeny here is the body size, so snout vent length of these different species, and the elevational range that they can occupy. So you can already see from this elevational range plot that there's been several different colonizations of high elevation environments on Sulawesi. And if we start to go through this phylogeny, we can see that early on in the evolution of these lineages, so 10 to 11 million years ago, we had these early east-west splits between these early paleo islands here that set the foundation for all these clays later on. So a lot of these clays that are east and west here remained on these eastern versus western paleo islands. So like western would be the yellow, orange, and this dark blue, and eastern would be this, or western would be the red, green, and light blue. So with a few exceptions, they pretty much remained on those different sides. And this is really due to kind of the proximity of these islands through time. So as we go through it, you see exceptional concordance with kind of these dispersal events and the predicted timing. So right at 8 million years ago, when this northern peninsula paleo island formed, we see 2 dispersal events to that island. Then as at 6 million years, when this second paleo island, northern peninsula paleo island formed, we see another dispersal there. And then at around 3 to 4 million years ago, as the mountains are starting to form on Sulawesi, we see this acceleration of diversification in this central core green clade. And this is associated with the uplift of the central core at that time. Finally, in the last phase, these lineages that are, as Sulawesi formed into its present configuration, there was some range expansion of these lineages that crossed into certain areas and expanded their distribution, but didn't speciate at that point. So very exciting to see this all kind of matching the timescales very nicely. And what's more amazing is that Skinks also displays a very remarkably similar pattern. So Skinks only really colonized these high elevation environments in one clade, we'll see here, but they show this kind of initial east-west splits at around 10 to 11 million years ago, around the same timing as the Sphenomorphous diversification. We see at 8 million years dispersal, three dispersals to this northern peninsula paleo island. And then later on at 4 million years, we see 2 dispersals to this tiny island offshore, Pelang, which formed around 4 million years ago. And then around the same time, we see this expansion and acceleration of diversification here in the central core, clade. So just kind of illustrate these patterns, so as the mountains formed, we see within area diversification increase. So if you look at these different kind of modeled biogeographic events, especially if you look at the within Paleo Island sympatric events, you can see a rapid increase in sympatric events here at 3 to 4 million years ago, or 3 to 5 million years ago here. So really highly tightly correlated with that period of mountain uplift on Sulawesi. And then along with that, we look at the number of lineages on each of the Paleo Islands. So the colors here correspond to the Paleo Islands. We also see around that same time bin a dramatic increase in the central core number of species in the central core. Sphenomorph is getting up to 14 or 15 species in just the central core. And Skinks is also showing a similar pattern. So really, the mountain uplift is tightly linked to this diversification, especially in the green central core, which is the area of the most kind of raised highland environments. So if we summarize this across like all the species Sphenomorphous across Sulawesi, and I'll just look at Sphenomorphous now because we have the best sampling for it, but we summarize all of it. We see that each mountain on Sulawesi developed a unique assemblage of elevationally stratified species. So most mountains have 5 to 7 species on them, and they're all coming from different clades. So the color of that and the letters on top correspond to the clade that they're in, and you can see each mountain has its own community there, and they all show some degree of elevational stratification. So we have elevation on the y-axis here, and you can see the elevational range that we sampled them. And we have species in low elevation up to high on every single mountain. And as we look at kind of how they've adapted to these high elevation environments, we see an increase in body size. So we've got the big fwens and the small fwens. They have an increase in body size that's correlated with elevation after accounting for the phylogenetic relationships. And there are a few exceptions. In Sphenomorphous, like you have this species here is Sphenomorphous Troponotus complex that has kind of a different. It's a very unique shape lizard. It's kind of a lowland generalist, that robust little chunky guy. But it seems to kind of be an outlier, and it is able to be large at low elevations. But generally, this pattern really holds, and it's very, very tightly correlated into the Skinks. So, why are they larger at high elevation? We don't really know, but it could be related to, thermal adaptation, being able to carry more heat for longer, or other things. But it's difficult to say without some more data. So how adaptive is this radiation? We know there are some adaptations happening, but what's really going on? So with the team of undergrads here, Allison, Anya, and Kayla, Anya's here today. We built this big morphometrics data set to look at kind of how the shape and size of these lizards change across the island. And I'll just talk about Sphenomorphous here, but I compared. I wanted to test if this rapid radiation in green here showed accelerated rates of morphological evolution, which would suggest that as they've rapidly accelerated in a number of species in the central core in this narrow area, they also adapted to new ecologies. I actually found that they don't seem to have it rapidly change morphologically during this rapid radiation here. So does that mean that this is not that there's no adaptation here? No, there's definitely some degree of body size and shape change, but it's not there's not, if they were really accelerated in morphological evolution, I would expect this clay to kind of be similar to the remainder. But there is a lot of kind of variation across the species in terms of ecology and other things that we've noticed in the field. Like we've noticed stream specialists down here. We've got like high elevation specialists, big and small ones throughout. We've got ones that we think might be semi-arboreal with long hind limbs. So there's definitely a lot of adaptive processes going on, but it's going to take a while to really sort this all out. So it's not as simple as these are the adaptive processes are causing speciation. It's probably happening in allopatry, and then maybe potentially there's some character displacement as they come back into the same regions. But this is definitely something I want to look into more. And the last little phase, last little thing that I did, which I will go through really quick here, is look at the thermoecophysiology, because I thought maybe they're adapting, morphologically, but also they might show different thermal tolerances, which could indicate that they're using different niches in these similar environments. So I, in the field, measured CT min and CT max with the Jesus fridge, the Lazarus fridge, that managed to survive an electrical issue with the generator. And but anyways, I measured this for several different species across different elevations on Gunung Dako, and I measured the CT min and CT max. So the performance of a lizard or an ectotherm is often shown like this. So thermal performance kind of varies with temperature. As the temperature goes up, they generally increase until it peaks and drops down. And the bounds of this curve are the critical thermal minimum and critical thermal maximum. We generally expect in thermal physiology that an ectotherm with like a higher peak might be a specialist, but it might have a narrower curve here. So it might not be, it's not able to occupy as many temperatures, but in a certain environment, it can have a higher performance. So from the literature on tropical mountains, thermal breadth usually increases with elevation. So they become more able to deal with cold temperatures. And this is mainly due to the evolution of CT min, which is directly correlated with the minimum environmental temperatures going up the mountain. That makes sense? So we expect an increase in breadth associated with CT min. So I'm going to show my data in a second, but I wanted to kind of frame it and compare it with the knoll lizards in Hispaniola, which has had quite a bit of thermal physiological research on them. And we see here that in the knoll lizards, as you move up elevation on the x-axis, CT max remains relatively stable, like I was just suggesting, and CT min slowly goes down to accommodate those colder temperatures on the mountain. And this leads to an increase in thermal breadth, generally linear increase in thermal breadth. So the knoll lizards are this classic adaptive radiation, right? But they all seem to kind of share this trend as they move up the mountains. When we look at the skinks on Gunung Dako, we see that CT max, again, relatively stable with a few exceptions. CT min significantly negatively correlated with elevation. And if we look at the breadth, we see that the breadth increases with elevation in most species, but there are these outlier species that have kind of a mostly stable CT min. It's kind of a almost flat line here. So these three species are Sphenomorphous that follow this curve. And this is the fourth species, Sphenomorphous, which has a really depressed thermal breadth. So I think this is an indication that in sympatry, this species might be more of a specialist versus this one is a generalist. And in Tithyscincus, which is the yellow, orange, and red here, we see an altogether different pattern where they all kind of have a stable thermal breadth going up the mountain, and we see potential kind of changes in the CT max as well. But for Tithyscincus, I have a really reduced sample size because they are hard to measure. So, there's limited what I can say there. But really exciting to see this. So this is evidence of something called thermal resource partitioning, which has been proposed to possibly explain other types of niche divergence in lizards and ectotherms. So if this is the case, then you could imagine across a mountain, there's all these different thermal niches that these lizards could occupy. So you could, up top, you could have a cold tolerant thermal specialist, versus lower down, you could have different types of the thermal ecophysiological specialists across the mountain. So this may be part of what explains the incredible diversity found even just on a single mountain on Sulawesi. So yeah, with that, I'll just summarize what I found here and today. First of all, that Sulawesi holds two incredibly diverse, elevationally stratified, and mostly undescribed skink radiations, each with around 30 or even more species. Both genera share these remarkably similar biogeographic histories, and overwater dispersal events are remarkably concordant with the origin of the Paleo Islands. The Paleo Island archipelago set the initial evolutionary foundation, and then mountain formation in the central core accelerated the diversification. And then species have adapted in many different axes, including size, shape, ecology, and possibly thermal niche partitioning. So this will be very interesting to go through in more detail down the road. So I'll be doing more formal species delimitation analyses and demographic analyses that in the future to kind of continue figuring out this radiation. So with that, yeah, that's the end. That's the end of my research talk, and I'll I know I'm running a little over, so if you need to go to, if you have something else going on, feel free to jump out. But I'm gonna mention a little bit of what I'm doing for my postdoc now, and so I'm now I've switched over to Ian Wang's lab in SBUM, and I'm looking, and with sponsors also Jonathan Richmond and Robert Fisher, I'm doing this NSF-funded project on the genetic basis of structural coloration in lizards and snakes. So the primary focus is this skink here in California, Plastiodon gilberti, which has these incredible red and blue tail color variation that's dispersed all over California. So I'm going to be doing genome scans, and this is related to the CCGP project that sequenced the genome of the species and also a ton of resequenced genomes across California. So I'll be including some additional samples and doing genome scans. I'll be doing RNA differential gene expression between the color morphs, and as they age, they lose their tail color, and looking at the uncolored adults, and then finally looking at the cellular anatomy to see, and the pigment cells themselves. And then I'm also kind of expanding this to other groups and looking at another Plastiodon in Florida that shows a similar red and blue variation. I'm also going to be looking at Eutropus rudis, which is a species I've been interested in a long time. And in Borneo and Sulawesi, I noticed it had this incredible polymorphism in the throat coloration in the males. So I want to see if that's a similar loci causing that, and then finally these amazing snakes in Indonesia, this Trimeresurus insularis, that have these blue, green, and yellow morphs, all can occur on a single island. So it's a pretty incredible system for looking at color evolution. So yeah, with that, I have a lot of acknowledgements, and I'm really happy to have done my PhD here, and I had an incredible committee with Jim, Rosie, Rory, and then on my dissertation committee, and Dave Wake for my quals committee. Yeah, thank you so much for all your helpful advice and feedback over the years. And then instead of going through all of these, I'm gonna show some photos, and so Jim, thanks so much for for being my advisor and all that you've taught me over the years. It's been amazing going to the field with you, and yeah, I really appreciate your desire and skills at developing these systems around the world that would not have been studied without a lot of groundwork out there. So I appreciate it. Also, thank you for helping me up the mountain, Jim and Jeff, when I dislocated my shoulder. Yeah, they literally had to haul me up this mountain. Like, I think Jim was on my holding my feet up, and Jeff was grabbing my one good hand while we climbed up this muddy slope. And that's why I was happy when I got out of here. So, but Jim, I had to kind of show this photo of Jim. Jim's not the only one, or Jim also has a occasional problem in the field. This is him with a foot infection, which I can only call trench foot, but where he's getting iodine put on it to kind of clear the infection. Yeah, I want to thank the McGuire Lab for just being a great group to be a part of, and all the nerd outs about herps that we had together, especially like Alexander and Umi for our first trip to Indonesia together. Yeah, all the greater MVZ community, it's just been a great formative foundational community for me. Been here a long time, and yeah, it's been really wonderful to be a part of it. Monica Albi was actually my first introduction to MVZ when she was running the prep lab. And then Carol, for all your help. Thank you for all your help with curating these specimens over the years. Lydia, for all your help in the genetics lab. Seriously, would not be possible without you. Yeah, and then Carla, Rory, and Michelle for help with getting me started in my research with the Sage Sparrow project during undergrad. These two, Mallory and Molly, you've been great friends and great support throughout my time here. It's been, yeah, I appreciate you. My master's advisors, Aaron and Todd. Yeah, it was really helped me develop into a scientist by doing that master's program and starting to work in Malaysia and Borneo. Yeah, I learned a lot from you, so thank you. All the friends from the Indonesian field work, just a selection here of great photos, but I think I promised someone I would never put this photo in a talk, but I couldn't help it. We are doing face masks at the end of a field trip, but yeah, Indonesian collaborators, we really appreciate all the help and all the time together in the field. In Malaysia, I've also had some amazing collaborators for field work there, and yeah, it's been really wonderful. The undergrads in the lab, thank you so much for all your help. It really got a ton of data, and yeah, would not have been possible to do these data sets without you. All my friends, local friends, and friends in the Bay Area and abroad that have just kept me grounded and kept me sane during this long period of time, or trying to study some random lizards, listening to all my talk about lizards. My family, my parents are here today, and yeah, you've been such a support, and yeah, always supported me kind of chasing after what I wanted to do, which ended up being very, very unique thing, but I've really loved it. So thank you. And my now extended family Kelsey and I just got married last summer, and yeah, my ever growing family has been an amazing support. And finally, Kelsey, thank you you've been the best support of all. You've you kept me grounded. You keep me kind of remembering why I do this, and I appreciate that so much. You're also my adventure buddy and my partner, so thank you so much. So with that, I will end there, and happy to take any questions. So